So as Amanda said, I'm going to be talking about the contributions of metabolism to the embryonic development of the Pacific purple sea urchin, which Latin name is Stronglyocentrotus purpuratus. And this is our an adult of our species of interest. So before I get started, I just wanted to quickly introduce myself. So yep, as everyone said, my name's Aidan. I live in Providence, and I'm working at Brown, and this is my research building. And I did my undergrad at Bard, and this was my old research building. So I've been in some very pretty science buildings, fortunately, lots of windows, very lucky. And while I was an undergrad at Bard, I did my senior thesis on the foraging behavior of woodpeckers. And then I did in a volunteer position in Rhode Island studying salt marsh sparrows. And then I took a complete left turn when the pandemic started because I wanted to move back to Providence and have a job. So I was just sort of looking for any kind of science job. And that's how I landed where I am today. And I live with my partner, Izzy, and our cat, Goji. And I recently attended the developmental biology of the sea urchin conference with my friend Cosmo. And this is us with our posters. And I presented on what I'll be talking about tonight, which is metabolic regulation. I wanted to quickly talk about why purple sea urchins are the model organism that I'm studying. An issue with some other model organisms is you have to wait a long time to get the materials to start your experiment. But with sea urchins, if you have an urchin, you pretty much have unlimited eggs or sperm. So you can do the experiments whenever you want, as long as you have animals. And they progress pretty rapidly through the embryonic stages, so we can watch them get to the gastrula stage, which is when they've already started to make a gut, which is only about 2 days into development, which is pretty cool. And they divide very synchronously. I don't know much about other model organisms, but they're all kind of at the same stage at the same time, which is very helpful if you want to get a lot of numbers in your data. Like if you're counting what they look like at a certain stage, you want them all to be at that stage. And urchins are very good at that. They're relatively easy to maintain in the lab. And I say relatively because they do still kind of die. And there's kind of nothing we can do about that. But in the wild, they live for hundreds of years, and you pretty much just throw them some kelp and they're fine. Another thing that's really important in developmental biology is that the embryos and the larvae are clear, which allows for easy visualization of fluorescent protein sensors, which I will talk about later. And sea urchins are part of the echinoderm family with other sea stars, sand dollars, and sea cucumbers. And they are an invertebrate, meaning they don't have a spine, but they are closely related to chordates, which give rise to vertebrates like humans. So as an invertebrate, they're slightly more related to humans than not. So they can be slightly applicable to human research as well. And a little bit more about the adults. Basically, the females give orange eggs and the males give white sperm. And something I really like about them is they have these like tube feet that they like wiggle around. It's not something I knew before I started working with them, but I think it's cool. So now I wanted to talk a bit about the developmental stages. And I think it'll help to have this understanding before we get to the second half of my presentation. So it all starts at fertilization, where these little specks are sperm and they're just kind of swimming around trying to find an egg. And this sperm found an egg right here, and they start to form what's called the fertilization envelope, which is sort of the transition from an egg to a zygote. It is now, a fertilized organism. And they stay in this fertilization envelope until they hatch at the morula or blastula stage, and they start swimming around. But while they're in the envelope, they're stationary. So they have a couple of symmetrical cell divisions, meaning all the cells are the same size up until the 8-cell stage. And then at the 16-cell stage, this is sort of the developmental time point that my lab is really interested in, because it begins the initiation of the animal to vegetal axis. And that's when you that's when the embryo is starting to decide which of these cells is going to go on to which cell fate at the end, like what organ are they going to contribute to. And another thing that's really interesting about the 16 cell stage is that there's an asymmetric cell division. So the cells at the animal hemisphere of the embryo divide evenly, but the cells at the vegetal hemisphere divide asymmetrically, which forms micromeres. And the micromeres sort of signify, like they are the first ones to get to be like committed to their cell fate. And I'm going to use these colors to track cell fates through the embryo, so you can really see like what they're contributing to. But the micromeres are going to contribute to the germline cells, which are going to be eggs and sperm in the adult, pigment cells, and the skeleton. The green cells here, their neighbors, are going to be forming the gut and like the mesoderm, which kind of just means like stuff in the middle. And then the blue cells here at the top are going to go on to make the ectoderm, or like kind of stuff on the outside. So yeah, and also it's been known for like hundreds of, or about 100 years, that the micromeres give a unknown signal to their neighboring cells, which tells them what fate they should become. And researchers found this out by implanting micromeres to the top of the embryo, and that embryo made two guts, which signified that it could tell these cells to have this fate too. So that's why it was known that the micromeres were the cause for them taking on this fate instead of the blue fate. So after the 16 cell stage, the embryo is just trying to make itself into a ball. And I sort of just colored in where the different cell fates are localizing. And then once they've fully made themselves into a ball, they've hatched, and they've really decided what's going to be inside the embryo and what's going to be outside of the embryo. They are like a unit, and they start to understand the localization of their own cells within the organism. So the ectodermal cells are starting to cover the whole outside, and the mesodermal cells are starting to form the gut. You can kind of see like this little protrusion. And then the micromere cells are also going to the inside of the blastula so that they can begin to form their structures. So this is called the blastula stage. And now we're moving to the gastrula stage, which means that they have a gut, which is formed by the green cells that we're going inside here. And basically, right now it's just a tube, and all they have is sort of like the butt end, and then they haven't opened their mouth yet, but they've started to make this tube. And the micromere descendants that come from these cells have started to form what's called skeletal spicules, which is just the beginning of the skeleton. And then these are the germline cells, which are going to go on to form eggs and sperm in the adult. And so moving on to the later gastric stage, this is the open mouth. So once the mouth opens, you can, there's another axis that you can sort of look at, which is oral to aboral. And then the structures are pretty much the same, but it just has an open mouth now. And so now the embryo has undergone most of its sort of cellular rearrangements. That's mostly what happens during gastrulation. All the structures have been formed. So now the embryo is just going to work on elongating itself. And that's what happens in the prism stage. It basically has all the same structures, but it's just sort of longer and pointier, because eventually it wants to become this very long pointy guy, which is called the pluteus. And here's where all the cell fates go in the pluteus. And something that I don't study, but I find very interesting, is the process of metamorphosis, which I will touch on in a second. But I also just wanted to show you guys like what this kind of map looks like in a publication. I find this to be more boring than what I drew. I tried to make it colorful and fun, but this is just another way to see sort of the same information that I was trying to present. So now I wanted to take a little tangent and talk about how does a pluteus become a juvenile urchin? Because I mean, the structures are just so different. And this is a process called metamorphosis. And so I just wanted to share a little video that I found from Notes from a California Naturalist, which is videos under a microscope of a pluteus actually, what it's doing is turning itself inside out, and the spines and the tube feet are just sort of reaching out from the inside, and it's kind of just like giving birth to an urchin. So right now we can just kind of see a couple spines. And then here we can see it's starting to reach out some tube feet, which is kind of how it's going to stick itself down somewhere until it's able to fully become a little juvenile that can walk around. And then here it's got a bunch more spines and it's got its tube feet. And also, if you guys have any questions about metamorphosis, I cannot help you. I'm so sorry. I find it very interesting, but it's something I just know nothing about. But I feel like it would be a shame to give a talk about urchins and not even talk about this crazy, weird thing that they do. Okay, so before I get back on track about what my research is actually about, I wanted to know if there were any questions. No questions?
It doesn't seem like anyone's put any in the chat yet.
Okay.
But again, anyone that is listening, you can type anything that you have questions about throughout. So it doesn't look like there's any yet, but I'm sure there will be as we go.
Yeah, I just wanted to try to make sure everybody could get the background before we moved on, because now I wanted to talk about more so what my lab studies, which is how does the egg that starts as just one cell become an embryo with all the different cell types, and then eventually a larva and a whole animal. And in my lab, our study really focuses on the micromeres. Everything comes back to the micromeres. In my lab meetings, my boss is like, but what about the micromeres? It's just, we love the micromeres. They're great. The micromeres teach us a lot about cell type differentiation. So I just wanted to go over the very basics of that. It is a very complex process. It involves a lot of different signaling pathways and a lot of different molecules that there's just so much research about it and people are still learning so much more about it. But you can track cell fate differentiation using cell fate markers. So here in pink, it's called VASA. It's a germline marker. And here you can see it's only really enriched in these micromere descendant cells. We're just a little bit past the micromere stage here. And that is one way that you can know that these cells are committed to becoming germline cells. And then here in green, we have something called beta-catenin, which is just a signifier that Wnt signaling is happening. It's a transcription factor, so it's just going on and activating genes that the Wnt signaling pathway wants to target. And the Wnt signaling pathway is important for cell fate differentiation. So it's basically just giving the cells information about where they are in the embryo. And what we like to study in our lab is what can we learn about cell fate differentiation by disturbing the process, which I will get back to sort of towards the end. And the way that we wanted to study cell fate differentiation was through metabolism and see if there were any differences in the different cell types in their metabolic processes. So there's sort of two main ways that a cell can get energy. Without oxygen, a normal sort of differentiated cell, meaning it has its fate, can go under glycolysis, which is pretty ineffective. It only really makes 2 molecules of ATP. But if you don't have oxygen, it's fine. About 30% of cells will sort of do that. But then when you have oxygen, the more preferred way to get energy is through oxidative phosphorylation, or later in the presentation, I'll kind of just abbreviate it to OXPHOS sometimes in my slides. And that makes you 36 molecules of ATP. So it's way more efficient. It occurs in the mitochondria, so it's why the mitochondria is called the powerhouse of the cell. It's making lots and lots of energy. It's very efficient. But what some people have noticed in proliferative tissue, which just means tissue that's dividing a lot, so embryos or tumors, regardless of whether they have oxygen or not, they're almost always going to undergo aerobic glycolysis, which just makes you 2 molecules of ATP, which is called the Warburg effect, because it was named after the dude who discovered it, whose name was Otto Warburg. And this is weird. And he was wondering why these cells that are trying to divide so much would choose such an ineffective way to get energy. And something about developmental biology is a lot of research about embryos can be applied to the study of cancer and tumors as well. So we were wondering, what are the metabolic processes happening in the sea urchin embryo? And my contribution to this research was confocal imaging. So basically, we're studying glycolysis and oxidative phosphorylation through 2 different sensors. The first one is a pyruvate sensor, and that is up here in green, and there's a membrane sensor in pink, just so you can get the shape of the embryo. And that sensor is called Pyronic SF. And here I'm showing the validation studies. So this is just our way of confirming that the sensors, for example, this pyruvate sensor, we needed to make sure that it was actually detecting pyruvate if we wanted to get any information about glycolysis using it. So here on the control without pyruvate, there's some signal, but then when you add pyruvate, it gets brighter. So that means that it is detecting pyruvate. But we wanted to get also a negative study as well. So we disturbed pyruvate kinase, which is an enzyme that makes pyruvate, and we impacted that through morpholino, which basically blocks the production of that protein. So if there's no pyruvate kinase, there's going to be less pyruvate, and the signal did decrease. So that's a good sign. Pyruvate sensor works. Great. And we did the same thing for the oxidative phosphorylation sensor, which is called GRX1 ROGFP2. I'm just going to call it GRX1. This sensor is a little bit different than Pyronic, and it's a ratiometric sensor. So it gives me 2 different readouts at different, sort of gets excited by lasers of different wavelengths. And so I take those 2 signals and I divide them by each other, and that's how I get the information about this sensor. So basically, that means that you'll see it as a rainbow. And so for the control, it sort of falls in the middle of this sort of readout color. But then when I add a reducing agent, the color gets cooler, which means that there's less oxidative phosphorylation happening. And when I add hydrogen peroxide, which is an oxidizing agent, we get to the warmer colors, which means that it's in its oxidized state. So that means that the oxidative phosphorylation sensor is also working. So now we can actually study normal development. So here we're looking at the pyruvate sensor, and we're looking at the pre-blastula stages. So we're looking basically from 16 cell stage until it's starting to make a ball. So initially, we see that the signal is enriched in the micromeres, and it's on the mitotic spindle, which is just like the division apparatus of the cell. And that was unexpected because glycolysis is expected to occur in the cytoplasm. We still haven't quite figured out why we saw this signal pattern, but we're going with it basically. But here you can see there is some signal here, but it's just very, very dim compared to later stages where even in non-micromere cells, the signal is much brighter and more visible. Initially, the micromeres seem to have higher glycolytic activity, but then eventually all of the other cells meet up and they all have the same, or give or take, the same levels of glycolytic activity. And here is the same sort of developmental phase, but we're looking at oxidative phosphorylation. So using GRX1, we see that oxidative phosphorylation is decreased in the micromeres. So that's the opposite of the pyruvate sensor. And then all of the cells slowly decrease in oxidative phosphorylation together, but it stays uniform. So we initially see asymmetry and then it all becomes the same. But the pattern is opposite to the pyruvate sensor, meaning that when glycolysis is up, oxidative phosphorylation is down, which is interesting. And it seems like early in development, since oxidative phosphorylation seems down, they might be relying more on glycolysis. So now we're going to look at glycolysis through gastrulation, which is the process of the cells ingressing and forming the gut. And it seems enriched in the ectodermal cells, and then it's most decreased in the cells that are committed to the germline fate, which again, I'm using the VASA marker. And there is the least amount of the pyruvate signal, which makes sense because these cells are actually in a resting state. And if they were not in a resting state, they would actually lose their VASA enrichment, and then the embryo would lose its germline fate, essentially. So it would not be able to form its germline cells. So these cells are the first cells to become committed to the germline, and they have to go basically to sleep to stay committed to the germline. But basically, pyruvate is increased in the ectoderm, decreased in the ingressing cells, and especially decreased in the germline. And now for oxidative phosphorylation through gastrulation, we see sort of, again, the opposite trend where it's decreased in the ectodermal cells, but it's increased in the ingressing cells. And so that's, again, it was interesting because it was the opposite. And now we're looking at the gastrula stage. For pyronic SF, the pyruvate sensor, we see that it's increased in the ectoderm and then decreased in the gut, and then even more decreased in the germline cells, again, because they're sleeping. And then for oxidative phosphorylation at the same cell stage, the germline cells, again, are very decreased. And that's shown by the co-expression with VASA to show that's exactly where the germline cells are. So it's most decreased here, and it stays increased in these gut cells as it was increased while they were ingressing. They stay increased, but the ectoderm is a little bit decreased compared to them. And I'm sorry if I went through that really, really fast, and I can go back later if anybody has any questions, but here's just the summary. Accompanied by a nice graphical abstract that my boss made. At the 16 cell stage, glycolysis is increased in the micromeres while there's decreased oxidative phosphorylation in the micromeres. And at the early blastula stage, there's sort of no regions of enrichment. All of the cells are sort of just doing the same thing. But then again, at the late blastula stage, we start to see some more asymmetry where glycolysis is decreased in the ingressing cells and even more decreased in the VASA-positive germline cells, whereas oxidative phosphorylation is actually increased in the ingressing cells. And at the gastrula stage, there's decreased glycolysis and oxidative phosphorylation in the VASA-positive cells, again, because they're sleeping, and there's decreased glycolysis in the archenteron, which is also the gut, just another like sort of fancy scientific word for it. And then in contrast, there's increased oxidative phosphorylation in the gut. So you can see all that information in this graphical abstract. So I won't repeat, but it's just sort of a visual way to see what I've been saying. So now that we sort of understand what's happening in normal development, we wanted to see if there were how inhibiting different parts of the different metabolic processes will impact development and the determination of cell fate. So why do we study inhibitors? Basically, it allows us to figure out which metabolic processes are crucial for development and which ones are maybe, yeah, they're kind of used, but they're not crucial. The embryo can still develop relatively normally. And also, are they important early in development or late in development, depending on when you treat with the drug or where you see a different looking embryo. And so the way that we chose to do inhibitor treatments allowed us to target micromere signaling or the potential micromere signaling, and I'll talk about that more on the next slide. But here I just wanted to quickly go over which drugs we're studying. Later I'm just gonna, I label them just based on the pathway that they inhibit, but here they also have names. But 2DG block glycolysis right here at the beginning. It prevents glucose from becoming glucose 6-phosphate, so it's right at the first step. And then the other drugs that we use block later in the pathway fatty acid synthesis. And we also block the electron transport chain, which is also known as oxidative phosphorylation. And this graph basically reveals what they did to the embryos. Like, you know, this drug caused gastrulation failure, but I'll show that in pictures as well. So we treated embryos only from 4 hours, which is when they look like this. They're at the 8-cell stage, and the cells at the vegetal end of the embryo are sort of starting to elongate to undergo that asymmetric division where they become a 16-cell stage embryo with micromeres. We treat only during micromere formation because this is when micromere signaling is suggested to be happening, when the micromeres might be talking to their neighboring cells and telling them to take on their specific cell fate. And then we look at them 2 days post-fertilization at the gastrula stage to see which cell lineages have been impacted. Were they able to make a gut? Do they have a skeleton? Are they even alive? And we call this process phenotyping. So what's the phenotype at day 2 when we treat only during micromere formation? So most of the metabolic inhibitors for this 30-minute treatment don't inhibit the process of making micromeres on its own, except for, it seems like, the oxidative phosphorylation sensor or oxidative phosphorylation inhibitor. But at day 2, there are significant developmental impediments by these inhibitors. So the control, you can see that it has a gut and it kind of has some skeleton. It's hard to see on Zoom, but this picture's fuzzier than I thought it would be. But inhibiting oxidative phosphorylation for this 30-minute period does not impact the formation of the gut. They still have a gut, they still have a skeleton, but glycolysis and, sorry, I think this one was meant to be fatty acid synthesis, not oxidative phosphorylation, but glycolysis and fatty acid synthesis seem to be the most important processes for development, because both of these inhibitors cause the embryo to either form no gut or die. And when the embryo is treated with the fatty acid synthesis inhibitors didn't die, they had a similar phenotype where they also had no gut. What's interesting about these inhibitors is you can also do a rescue where, so for example, with the glycolysis inhibitor, if you don't have a rescue, again, there's no gut, but if you add glucose about an hour before adding the drug, or like 30 minutes before adding the drug, then the embryo is able to sort of combat the impacts of the inhibitor, meaning that the inhibitors are specifically targeting glycolysis, and glycolysis is the reason for, or blocking glycolysis is the reason for this phenotype. It's not some other sort of off-target effect. And the same goes for when you inhibit fatty acid synthesis. If you treat the embryos with a fatty acid, they are able to form into normal embryos. And again, here's just the phenotypes without the rescue again. And so we also took a look at cell fate determinants that I showed earlier, and to see if these inhibitors had an impact, because they did seem to be impacting the process of cell fate determination, because again, these embryos weren't forming a gut. So how does a 30-minute treatment with a drug, no phenotype at the 16 cell stage, like what's happening that's impacting gastrulation by day 2? So we looked at some metabolic inhibitors and their impacts on cell fate markers. So over here in this panel, we're looking again at VASA, the germline marker in pink, and beta-catenin in green, which is the Wnt signaling marker that's important to have only in the vegetal hemisphere, and that lets those cells know where they're located basically in the embryo. So in the control, VASA is enriched in the micromeres, or the micromere descendants. Beta-catenin is enriched only in this bottom half of the embryo. When you treat with the oxidative phosphorylation inhibitor, the process goes on relatively normally. But when you inhibit glycolysis, you lose the VASA enrichment. It sort of just becomes equally dispersed across all cells. And the same thing happens with the beta-catenin. So basically, when you block glycolysis, you block the ability for the cells to determine these fates. And something similar happens when you inhibit fatty acid synthesis. So it suggests that these cell fate markers might be sort of the reason why these inhibitors are having their effect, is because they're also impacting the process of this patterning. And so what does this mean? So these results imply that the fatty acid synthesis and glycolysis are required in early development, but not as much oxidative phosphorylation, because the oxidative phosphorylation sensor doesn't cause a phenotype at day 2. And it also suggests that the proper metabolic regulation may be involved in properly distributing the cell fate factors, like we talked about VASA and beta-catenin. And from the results of my confocal imaging of normal development, initially embryos seem to rely on glycolysis, and then oxidative phosphorylation sort of takes over later once they've more so determined their fate. And that is my whole talk, and I'd be happy to take any questions.
Thank you, Aidan. That was wonderful. So let's see. Let's look at the chat. We've got one question here. So looking at your research, how do you think this is going to impact other areas of sea urchin research and biology as a whole?
Interesting. So invertebrate research is almost always sort of compared to like vertebrate research. So it'll definitely impact vertebrate studies, which might also basically things can be compared, but also these experiments will most likely be repeated in a vertebrate model to see if they have similar effects, or maybe to see what the differences between invertebrates and vertebrates are. And also, this helps us understand more about how cells are determining their fate during embryogenesis, or the process of a developing embryo. So here we've kind of shown that metabolism is an important aspect of that developmental determination.
Wonderful. Okay. See here, Wallace, did you have any that you're looking at?
Yeah. So you were talking about how certain germlines, they had this determination, the fate. Is it always that certain germlines are determined first, or do other germlines sometimes, when you were talking about VASA?
Yeah. So the germline is like one fate, and like becoming the gut is a separate fate, and like becoming the ectoderm is a different fate. So basically, the germline is the first to determine its fate, and we can see that because it's one of the first markers that you can look at. Like it's enriched as soon as the micromeres are formed, whereas here we're looking a little bit past the micromere stage because beta catenin only sort of shows this pattern a little bit later. So basically, you can look at, there's like, all these different pathways involved, and you can look at different markers for these pathways to look for markers for other cell lineages. But it's a little later in development because the micromeres are sort of fast to determine their fate.
Great, thank you.
And going back to your very first slide with those beautiful images of the sea urchins,
Yeah.
I know myself, Wallace, and I'm sure many others are probably wondering, is that cowboy hat real? And why do sea urchins love to have things on top of them?
So this is not my picture, but again, I didn't want to leave this out of my sea urchin presentation because, I mean, this is awesome. This is great. It is a real cowboy hat, and sea urchins like feel really safe when they have something on top of their head. And in the aquarium, they like to be in the dark. It just makes them feel safe, and yeah, they just like to be a little bit covered up. So people who have sea urchins as pets, if you have a little cowboy hat, you can just put it on your little guy.
That's amazing. I
Yeah.
love that fact. Does anyone else have any questions? Any of our other attendees? You can use the chat feature now if you have any questions for Aidan. I'm going to give everyone a second just to type anything in if they need to.
I believe too, if you would rather speak your question instead of typing it, you can ask to be unmuted, and Amanda and I will unmute you and you can ask your question.
I can also do a little show and tell while we're waiting. I have some sea urchin shells. So this is a sea urchin that's more closely related to the species that I was studying. So this is a sea urchin that's been dissected. So my friend Cosmo, who's actually in the chat, dissected this guy to take out the ovaries or testes. And so that's why it keeps its spines. If an urchin dies naturally, then the shell, the spines will fall off the shell, and it'll be this sort of pretty shell. And this is sort of a more distant relative of the sea urchin that I've been studying. This one's a pencil urchin. And he just looks more prehistoric, I think.
Awesome. So I have a question for you as well.
Yeah.
So you've been studying sea urchins for a little while now, but do you think moving forward with your research, is sea urchin something that you really want to focus on long term? Or do you have any other areas of developmental biology that you're really interested in?
Yeah, so I started studying ecology at Bard and studying birds, and I would definitely like to get back into field work. So sort of get away from developmental biology completely. I think it's obviously been really interesting to learn about, but sort of not my cup of tea for the long term. So yeah, I'll end up moving back to studying birds when I can.
Wonderful. Thank you so much, Aidan.
Yeah.
So if there are no more questions now, that's totally fine. So for all of our attendees, if you think of questions later and you're like, oh, I really wish I should have asked Aidan that in the presentation, you can always email education@starhop.com. Those will go directly to me and Wallace, so we can relay any questions that you have and get you answers as soon as we can. But I want to thank you, Aidan, so much for coming on and joining us today and teaching us
Yeah, of
all
course.
about.
Thanks for having me.
This is really wonderful. But I think this is just about the end of our presentation here. So I hope that everyone has a very wonderful evening and a great weekend, Mother's Day, if you are a mom or celebrate with your mom. And thank you so much. So have a wonderful night.